Good afternoon, everyone. Thank you to the organizing committee for inviting me to give a talk in this very informative, I'm sure, webinar. And first of all, also salute and thank you, especially to the medical colleagues and clinicians who are frontliners in our concerted effort to battle against the COVID-19. So this is the title of my talk, which has been introduced by Prof. Karim. And this slide shows the contents of my talk today. So I will begin first with a very brief introduction to the SARSCoV2 structure. So coronaviruses belong to the Coronaviridae family of the order of Nidovirales. And they are actually a group of enveloped viruses having a positive single-stranded RNA genome, and they are mostly pathogenic. The COVID-19, so we know that it's caused by the SARS-CoV2, and it is a more pathogenic form in comparison to the previously identified SARS-CoV, which had a massive outbreak in Hong Kong, and then the Middle East Respiratory Syndrome coronavirus, or MERS-CoV. In terms of its structure, SARS-CoV-2 contains 4 structural proteins: the spike protein, membrane protein, envelope protein, and nucleoprotein. And inside we have the single-stranded positive strand ribonucleic acid, or RNA. So we may ask the question of how does the SARS-CoV-2 virus attach to and infect the human cells? And I'm sure most of you already know that it binds to the host cell surface receptor known as the angiotensin converting enzyme, or ACE2 receptor, through the spike proteins on its envelope. Since the lung cells are known to have an abundance of the ACE2 receptors, so it is no surprise that they are the main target of attack by these viruses. Now, so coronaviruses actually have the largest RNA viral genome, with their genome sizes ranging from 26 to 32 kilobases in length. SARS-CoV-2 has a total genome size of about 29,900 nucleotides, or 29.9 kb. So the genome structure of the SARS-CoV-2 is shown in this diagram. It has 11 protein coding genes with 12 expressed proteins. So the open reading frames, or ORFs, are arranged as in the order of replicase and protease 1A, 1B, and then followed by the major structural proteins, the S, E, M, and N proteins. And these are considered as the major drug or vaccine targets. So the SARS-CoV-2 genome share about 82% identity in terms of its sequence with the SARS-CoV and the MERS-CoV genome, and more than 90% sequence identity for the essential enzymes and the structural proteins. So the high level of sequence similarity between the SARS-CoV-2 genome and the other coronaviruses revealed a common pathogenesis mechanism among these coronaviruses. In addition, it also has important implications for designing nucleic acid-based diagnostics. So, as of now, there are already hundreds, if not thousands, of the genome sequences of SARS-CoV-2 that have been released by different labs around the world. Identifying unique regions of the virus genome and conserved regions of the virus is crucial for development of laboratory molecular diagnostic tests. So through the sequencing of the genomes, it has been revealed that SARS-CoV-2 is closely related to the bat coronaviruses with a similarity of around 80 over percent with these bat coronaviruses. And the E gene has been shown to be the most conserved region with a similarity of more than 93%, followed by the N and the N gene regions. Whereas the sequence identity of the ORF1A is greater than that of the ORF1B. And the S gene, notably, exhibits the lowest similarity, about approximately 75%. So the ability to diagnose the SARS-CoV-2 reliably would facilitate the identification of the infected patients. And so development of a reliable assay has been one of the foremost matters for public health as well as for clinical interventions. So we have molecular or nucleic acid-based assays as well as serology assays, but currently nucleic acid assays by reverse transcription real-time RT-PCR is the main approach for diagnostics. For RT-PCR assays, which specific genes are selected is really crucial. So the E gene has been shown to be highly similar to that of other coronaviruses, a fact that can mean that it can serve as the first-line screening tool. In addition, the ORF1B, which involves the RDRP gene, the N and S genes, they are relatively specific, so they are so-called the unique genes that are worth targeting when we do the RT-PCR assays. In clinical RT-PCR protocols, single target versus multi-target assays are a thing that we have to consider. For single target, it refers to the use of one set of primers and related probes to target a single gene region, whereas the latter uses multiple sets of primers and also their related probes, such as reagents that target the ORF1AB and also the N genes, which are used in China, and those that target the N genes, as well as the E gene or another set of gene in Thailand. So since SARS-CoV-2 is a single-stranded RNA that is prone to mutation, increasing the number of specific targets in the RT-PCR system will increase the testing sensitivity. However, if the primers are not specifically designed, multiple targets in the PCR system could interfere with each other, thereby reducing the amplification efficiency and also the clinical sensitivity. So therefore, selecting an appropriate number of targets for the RT-PCR protocols depends on the purpose of the detection and the specific primer sequence, and as well as the results of the performance verification. So at the beginning of the outbreak, WHO has supported access to COVID-19 in-house PCR protocols by posting them online on their website. So many in-house assays have been developed by the various institutions in different countries, such as the China CDC, the Institut Pasteur in France, the US CDC, and so on. So This table summarizes the available in-house protocols developed by these various institutions. However, WHO stressed that being listed in this document does not imply any endorsement or validation by the WHO. And as you can see, these different protocols target different selected genes of the SARS-CoV-2 virus. And these slides show the key points from the WHO's interim guidance for laboratory testing for the SARS-CoV-2 in suspected human cases. So rapid collection and testing of appropriate specimens from suspected cases is a priority, and this should be guided by a laboratory expert. Respiratory materials such as the nasopharyngeal and also the oropharyngeal swab in ambulatory patients and sputum if it is produced, and or endotracheal samples or bronchoalveolar lavage fluids or BALF in patients with more severe disease are usually used. As extensive testing is still needed to confirm the 2019 or the SARS-CoV-2 pandemic, and the role of mixed infection has not been verified, multiple tests, including serology and also radiology, may need to be performed, and so sampling sufficient clinical material is recommended. So for the nucleic acid testing, since 4 different human coronaviruses are endemic, so the amplification and detection of SARS-CoV-2 specific sequences can be diagnostic without the need for DNA sequencing. So it all depends on the design of specific primers and the unique sequences that are targeted to the virus itself in the RT-PCR system. So this slide is a schematic of the process flow for RT-PCR testing. First step is the sample collection. So nasopharyngeal swab or NPS is the sample of choice in most labs. And step 2 is the extraction of the RNA of the virus from the samples that are collected. So there are different methods of the RNA extraction. In some RT-PCR kits, it is a straightforward lysis of the samples using some buffers, which takes about 30 minutes. But some labs prefer to use a more complicated or more extensive kit for nucleic acid extraction, which could take up to 2 hours, but it produces a more pure RNA sample. In the third step, this is actually the RT-PCR step, where the reactions are set up and then placed into the real-time PCR machine. So reaction setup itself may take between 1 to 2 hours, depending on the number of samples. And then the actual PCR run in the machine takes about 2 hours. So these RT-PCR depends on the fluorescent detection of the amplified nucleic acids using the specific genes of the primers. The last step is actually where the results are compiled and analyzed, and if necessary, tests may be repeated. Next, I just want to talk about the type of samples, and that type of samples do matter. So as I mentioned in the previous slide, there are different types of samples that are used in different settings and different labs. So the type of sample actually play a big role in the ability to produce a positive result. And secondly, for the molecular assays, the selection of specimens is crucial. So it has been found that viral loads of respiratory tract specimens are highest in the BALF, followed by sputum, and then nasal swabs and pharyngeal swabs. So if situation allows, the sample selection should be prioritized in the same order to prevent misdiagnosis caused by insufficient viral load. Several studies have confirmed the validity of this selection priority. For example, Zhou et al. Found the positive detection rate of the nasopharyngeal swabs in 14 out of 18 patients was higher than that of oropharyngeal swabs. And the PCR CT values is the cycle threshold values of the former were also lower. The lower the CT values, that means the higher the viral load. In addition, a recent study by Wang and colleagues evaluated the positive detection rate of over 1,000 different specimens, and their results revealed that the positive rate of BALF was highest, followed by sputum, and then nasal swabs, throat swabs, stool samples, and lastly, blood and urine samples. They found 0% positive rate. So therefore, if multiple sampling methods can be adopted for patients, specimens with high positive rates such as BALF or sputum should be selected preferentially. Having said that, in clinical application, collecting BALF requires a complicated procedure, so it is not suitable for all patients. In addition, many patients have non-productive cough, so the feasibility of using sputum is also quite low. Therefore, the nasopharyngeal swab and oropharyngeal swabs still serve as the main sample types for clinical testing. But we have to bear in mind that these samples may yield negative results on molecular assays due to the low viral loads, and it could lead to misdiagnosis. So when it comes to the interpretation of the RT-PCR test, firstly, diagnosis of the infection mainly depends on positive viral nucleic acid test results and not on the clinical symptoms. And secondly, asymptomatic individuals may get a positive viral nucleic acid RT-PCR. So these asymptomatic infections that yield positive viral nucleic acid results may be potential sources of infection. So even if results from the NPS or the oral swabs are negative, the virus may also harbor in the gut, or the recovered patient may still be a virus carrier. So all these things should be taken into account when interpreting the results. Therefore, looking at the clinical symptoms is very important. Fourth, sampling of different sites in suspected patients or repeated sampling at different infected stages may prevent false negative results from insufficient viral loads. So the WHO interim guidelines has a very clear statement here, right, regarding negative test result. So we have to be cautious when interpreting negative test results. So there are at least over 50 different RT-PCR tests that have been developed for the COVID-19 testing so far. Not only in-house tests, but the different CDCs of different countries, but also by different commercial companies. And they target different genes. And also we can see here in the table, the sensitivity of the infection, in terms of the limit of detection, also varies quite widely, in 10 to different to even 100-fold in terms of limit of detection. So yeah, this is just continuation of the different RT-PCR tests that are available. So I'm sure a question that now you will ask is, how accurate are these RT-PCR tests? So to evaluate the sensitivity and specificity, or you could call it the accuracy of these tests, actually we need to compare these tests with a gold standard test. Unfortunately, to date, there is a lack of such a clear-cut gold standard for the COVID-19 RT-PCR test. So a systematic review of COVID-19 tests reported false negative results of 2% to 29%. So this is equivalent to a sensitivity of about 71 to 98% based on negative RT-PCR tests which were positive on repeat testing. Since the current studies show marked variation between the different tests, and they are likely to overestimate the sensitivity, so it is practical to use the lower end of the current estimates from different systematic reviews with the approximate number of 70% for sensitivity and 95% for specificity. So you have heard me talk about viral load, and also you may have also seen in different publications. So viral load, in terms of the layman understanding, is total amount of virus that a person has inside them. But for the virologists, viral load is a specific term in medical virology which usually refers to the quantity of measurable virus in a standard volume of material, which is usually the blood or plasma. So it is usually measured in terms of number of virus particles per ml of blood. So we can measure it in terms of relative amount of virus or absolute amount of virus. So the outcome of infection, whether you get severely ill or only get a mild cold, can sometimes be determined by how much virus actually you have in your body. So in terms of virus shedding, this term refers to the release of new viruses into the excreta, which is including the respiratory secretions, like when you sneeze or cough, your sputum, saliva, urine, feces, and etc. And this is once the virus have successfully replicated inside the cells. For COVID-19, early reports from China suggest that the viral load is higher in patients with more severe disease, which is also the case for SARS and also influenza. And the amount of virus in different nose swab samples can vary over a 1 million-fold range. This is probably influenced by the stage of the disease, the efficiency with which the infection has colonized the patient at the time of doing the sampling, as well as the amount of nasal swab that the nurse or the medical personnel take from the swab. So serology assays have a value as well in terms of diagnostics. And the aim of serology tests is to detect individuals who have had previous infection and therefore theoretically have developed the immunity. So serology tests are tests which detects antibodies, which is the immunoglobulins, or in short IG, in the blood or the serum. So the time course and the accuracy of serology tests are still being debated and are being studied. But the same principles of incorporating the serology test result with other tests and also with the clinical findings is still apply as with other type of tests. So in SARS-CoV-2 infection, the RBD, S, and the N proteins are the main antigens which stimulate the immune response and produce the IgA, IgM, and IgG antibodies. So these are the proteins that serology tests usually target. IgM usually indicates the acute infectious stage. So they will be the first type of antibody that you see, while IgG represents the middle and later stages of infection or even the recovered infection. So you will see it later. Whereas secretory IgA are usually indicate the or represent the mucosal immune response. So application of serology assays, it can be used in the diagnosis of suspected cases with negative RT-PCR results, either in conjunction with molecular tests or additional testing. And they do have the potential value of antibody titers for evaluating the prognosis and also the recovery of patients. We know that the antibody titer in most patients increases around 10 days after symptom onset, although this will vary from individual to individual. Serology test is also useful for surveillance or public health in terms of monitoring the community. So this slide shows some of the serologic tests which are recommended by FDA. So this is a continuation. So in terms of the serology tests, there are less tests compared to the RT-PCR tests that have been developed so far. So cross-reactivity with other subtypes of coronaviruses may interfere with the findings of serology tests. And also for antibody assays, it is susceptible to the influence of endogenous interference, including like rheumatoid factors, heterophilic antibodies, and also the complements. Not only that, the exogenous factors like specimen hemolysis, if the specimens have lysed already, and this also will produce false positive results. So the sensitivity and the specificity of serology tests are not as high as the molecular nucleic acid based tests or RT-PCR tests, but they do have the value as well. Next, I'll talk about the radiology assays or tests. So WHO has actually listed several scenarios whereby radiology tests, specifically chest imaging, should or should not be used. So chest imaging has been considered as part of the diagnostic workup of patients with suspected or probable COVID-19 disease where the RT-PCR is not available or where the results are delayed or are initially negative in the presence of symptoms suggestive of COVID-19. Imaging has also been considered to complement clinical evaluation and also the laboratory parameters in the management of patients already diagnosed with COVID-19. And in China, I noticed that they combine the RT-PCR test with CT scans and ultrasound results for a more comprehensive picture of the findings for patients. So this one is a slide about the biomarkers that are important for COVID-19. Dr. Farzana has already introduced to you some of these important biomarkers. The C-reactive protein or CRP, which is a plasma protein produced by the liver and induced by various inflammatory mediators such as IL-6. Although C-reactive protein is not a specific marker, clinically we have seen in different retrospective studies that showed that majority of patients in the severe cohort will have significantly higher levels of CRP compared to the non-severe cohort. Another marker, which is the serum amyloid A or SAA, which is also another acute phase marker. This is responsive to both viral and bacterial infections compared to CRP. So it may complicate the interpretation of result if the patient has pneumonia of bacterial origin. Now, interleukin-6, IL-6 is a cytokine released by macrophages which rise sharply in severe manifestations of COVID-19. And Dr. Farzana also has mentioned about cytokine storm, and IL-6 is one of the key mediators in the cytokine storm. The other one is the white blood cell count, specifically the lymphocytes, where the ratio of neutrophils to lymphocytes have been indicated to be a potential biomarker for early detection of severe COVID-19. D-dimer, this D-dimer originates from the lysis of cross-linked fibrin with rising levels and indicate the activation of coagulation and fibrinolysis. So early studies have associated COVID-19 with hemostatic abnormalities, with some studies observing elevators of D-dimer in the non-survivors compared to the survivors of COVID-19. Besides that, we also have LDH or lactate dehydrogenase, which I didn't put here. LDH levels were correlated with CT scans actually, and it shows a significantly higher level in severe pneumonia of COVID-19. Platelet counts also. Platelet counts in some meta-analysis, it's been revealed that those with severe COVID-19 infections had significantly lower platelet counts. And when using mortality as an endpoint, non-survivors had significantly lower platelet count.
Excuse me, Prof. Chong.
Yes.
You are exceeding the time. I'm sorry.
Oh, I see the time. Sorry, so sorry.
How many slides?
This is my conclusion. Yeah, so it's
Yeah.
just a summary
Okay.
of the slides that.
Thank you for your cooperation.
Thank you, thank you. Thanks, Prof. Karim. So, in conclusion, accurate diagnostic assays are crucial for identifying the infected persons, and RT-PCR is the widely employed as the confirmatory molecular testing, which confirms active infection, whereas serology can be a complementary test that that confirms either mid to late stage of infection. Chest imaging has value in complementing other modalities of testing, and biomarkers can be used in conjunction with chest imaging for hospitalized patients and can aid in clinical decisions for triaging patients for clinical management. So thank you everyone for your kind attention.